SmaXtec Animal Care is a company based in Graz, Austria, and is the worldwide market leader for intraruminal sensor systems for dairy cows. We are developing and distributing these sensors which are used inside the cow's rumen to monitor animal health and certain nutritional parameters. The measurement and interpretation of pH values in the cow's rumen is one of the unique selling propositions of SmaXtec. The project's main objective was the successful establishment for a more durable alternative to the probes currently used in our sensor units.
Our role at RISE, Research Institute of Sweden, was to characterize the pH probe prototypes and, based on the results, to give recommendations for possible improvements based on our expertise in sensor development and electrochemical characterization. The characterization involved the evaluation of signal uniformity for pH values from pH 4 to pH 10, the influence of the working electrode on sensitivity, reference electrode performance and response time, and the dependence of the sensor response on the actual buffer chemistry. According to the results of characterization, it can be said the service lifetime of the probes will likely exceed the durability of available commercial solutions. We have observed that the response varied somehow with the buffers used. However, this is of minor importance for the present application, since the probes will be used in a single environment.
The development of these much more durable pH sensing probes was the primary goal of the project with SmaXtec. Apart from producing these laboratory demonstrators, Joanneum Research has also elaborated procedures that are reproducible and practicable for the fabrication of pH sensing probes that are compatible with the SmaXtec bolus sensing hardware.
The characterization results showed remarkable uniformity across the range of the tested pH sensors. The manufacturing of the working electrode does not influence the sensitivity, and sensor responses to pH changes were linear in the tested pH range.
During the development, we had to go at some chemical and physical fundamentals, as well as doing, let's say, pure engineering. And at the end, we have now prototypes that are ready for installation into the commercialized SmaXtec hardware, and that are ready for field testing in cattle rumen to prove the service life of more than 18 months as compared to about 6 months of the pH sensors that are presently used in the commercialized SmaXtec hardware.
The project result shows a pathway to significant improvement of sensor durability and yield in production. Less sensors need to be used and far more available insights with the same numbers of sensors. We were overwhelmed by the expertise and the commitment of the scientists involved in the project. Besides the actual project results, it was a great opportunity to get to know them, their way of working and how we could work together in the future.